Alright, welcome back to the second half of Chapter 9, or second video, I should say. In this video, we'll be talking about examples of cancer-causing viruses, and they get organized into these four classes. So retroviruses, RNA tumor viruses, small DNA tumor viruses, and large DNA tumor viruses. And as you can see, so these are all the known cancer-causing viruses in humans. There's a relatively small handful, but as we discussed in the beginning of the chapter, they are responsible for a pretty substantial number of cancer cases worldwide. So what I want to do is just go through these one at a time and at least give you a kind of a brief overview of what kind of cancers they're linked to and any known mechanisms in terms of how they're believed to be tied in with cancer. So just generally speaking, although these different oncogenic viruses are unrelated to each other for the most part, they do all target similar pathways that we talked about in the first part of the chapter, things that control cell proliferation or, limit too much cell division. Or they will be involved in evading immune responses, which can lead to essentially out of control virus replication and chronic inflammation over time. And as we'll discuss, that chronic inflammation is often linked to increased cancer risk as well. So for any of these viruses, we could say that they either directly or indirectly contribute to development of cancer. And if it's direct, we would say the virus actually encodes some proteins in its genome that help drive the formation of cancer, whereas indirect would be more the chronic inflammation type route that I mentioned. And really for all of these as well, you could consider the development of cancer just to be a byproduct of the infection. So, we don't really know the motives of the virus, but we do know viruses, one goal seems to be just to make copies of themselves. So the cancer is believed to be just a sort of an unintended consequence. Although, if you are a virus and the one thing you need is host cells to be able to propagate yourself, if you do happen to cause increased cell division, right, and the formation of new cells, that could actually be advantageous to you. Okay, so as I mentioned, we have these four broad categories, and the first one is the retroviruses, and we just have one example, which is HTLV1, human T-cell lymphotropic virus. And before I get into its specific cancer-causing mechanisms, there is actually a pretty rich history of understanding how retroviruses can be linked to cancer, and this goes all the way back to the early 1900s. Peyton Rouse's discovery of Rouse sarcoma virus, which was a cancer-causing virus in chickens. And keep in mind, retroviruses, right, they have an RNA genome. They go through this reverse transcription process where they make a DNA copy of their RNA genome, and then that DNA copy integrates into the host chromosome. So this discovery by Rouse was a huge deal because it was the first link between viruses and cancer, and he and many others believed at that time that maybe all cancers were caused by viruses, which we now know to be incorrect. But it kind of set off a bit of panic, because if that's the case, then maybe you really could kind of catch cancer, right? You could get it from these contagious agents. In more modern times, with the advantage of being able to do DNA sequencing, researchers were comparing two different strains of Rouse sarcoma virus. One that seemed really benign, actually did not cause cancer in chickens, versus one that did, on the bottom here. And what they found was really the only difference was the cancer-causing virus contained this additional gene, which is called a viral oncogene. And of course, there was then great interest in, okay, well, what is that oncogene? How does it work? How did the virus get it? Why do some strains have it and some don't? And what they found basically was in the case of Rous sarcomavirus, this gene came to be known as SRC. Its function was found to be a kinase enzyme, right? Phosphorylates other molecules in the cell. But interestingly, they found the SRC gene was present in the chicken genome. And in fact, it seemed to be present in lots of animals. So we see it in chickens, we see it in mice, we see it in humans even. And we have since found examples of a number of other different retroviruses that also contain these host cell oncogenes in their genome. And the idea is these viruses picked up the oncogene from the cells that they infected. And it's believed they do that by when they integrate into the host cell chromosome. If the virus happens to integrate right next to a proto-oncogene, like SRC, for example, when the viral genome gets transcribed, it may also transcribe that SRC gene that's sitting right there next to it, and that may become incorporated into its genome. And we call these viruses that contain oncogenes acute transforming viruses because they're so adept at causing cancer. The presence of those oncogenes really drives a quick transformation. Excuse me, had to take a little drink there. Catch in my throat. So those acute transforming retroviruses are in contrast to non-acute or slow transforming retroviruses, which is what HTLV1 would fall into this category. But they don't contain oncogenes, but rather the vast majority of viruses in this category are believed to cause cancer through insertional mutagenesis. Through this process that would look something like this. Could be insertional activation or inactivation. Here's an example of activation. So in this case, the viral DNA inserts relatively close to this proto-oncogene in the host cell chromosome. This is the MYC proto-oncogene in this case. And what happens is because the viral genes get transcribed at such a very high rate, some of that excess transcription can rub off on neighboring host genes. So for example, the MYC gene that's normally expressed at a relatively small amount gets overexpressed, right? Its transcription gets ramped way up. And so now we're making too much of this proto-oncogene that helps promote cell division, and it's helping to encourage that cancerous state. Kind of the opposite situation would be, well, imagine what if this viral DNA inserted right into the middle of a tumor suppressor gene in the host chromosome. It would basically mutate, break that tumor suppressor gene, and we're effectively cutting the brakes, right, on regulation of the cell cycle. So through either way, insertional activation or inactivation could be a problem with these retroviruses. And really with any type of virus that integrates into a host chromosome. So after having said all that, the HTLV1, our human example here, actually isn't thought to cause cancer primarily by either of those mechanisms. It's kind of unique in the world of retroviruses in that it encodes this TAX gene, which is a transcription factor that can interfere with the retinoblastoma tumor suppressor protein. But it doesn't seem to have a human counterpart, right? So it seems to be unique to the virus genome. And in terms of this particular virus, it's a relatively small percentage of those infected that actually develop cancer, and it seems to only happen after many years of infection, so 20 to 30 years. So it's a relatively rare occurrence, but it can cause this very aggressive T-cell leukemia. All right, our second group is the RNA tumor viruses, which is in humans so far just hepatitis C that's linked to cancer. Hep C is a positive sense, single-stranded RNA virus, Flaviviridae family. when you think hepatitis, you probably think liver. So it shouldn't be surprising that Hep C primarily infects hepatocytes, which are those liver cells. And a relatively high percentage of Hep C infections become chronic. The immune system just can't completely clear them, so the virus hangs around for many, many years. And a high percentage of these chronic HCV cases are linked to a common liver cancer called hepatocellular carcinoma, or just HCC, we'll call it. So in the U.S., there's thought to be several million chronic HCV cases, and about half of the HCC cancers are linked to this particular virus. This is HCC is a cancer you don't want to get, so it's got a very low 5-year survival rate, less than 12%, thought to be the second leading cause of cancer-related mortality. You can see a picture here of a woman with very advanced HCC, very distended abdomen. It looks pretty horrible. And so, really, there's two ways HCV can contribute to liver cancer. There's indirect mechanism, which, as I mentioned, is chronic inflammation, long-term inflammation. So, of course, that can cause scarring of the liver, which is cirrhosis. But long-term inflammation also leads to the production of lots of reactive oxygen species, which can easily damage DNA and cause accumulation of additional mutations in oncogenes and tumor suppressor genes in those infected cells. It can also contribute to HCC by direct mechanisms through the role of these 4 different proteins encoded by HCV. So HCV genome codes 10 proteins in total. 4 of those have been linked to cancer. There's the HCV core protein, which has, as you can see, a number of different effects. Can promote overexpression of cyclins and CDKs, increased expression of telomerase, inhibits apoptosis. All of those would be bad in relation to cancer. This NS5A, which again inhibits apoptosis. NS5B interferes with retinoblastoma protein. NS3 mechanism seems unclear, but it has been directly linked to cancer as well. So HCV's got a few different arrows in its quiver there, direct, indirect mechanisms. Then that leads us to the small DNA tumor viruses, which include hepatitis B, the Merkel cell polyomaviruses, and HPV. So Hep B does share some similarities with HCV. Of course, preferentially infects liver cells. It's linked to chronic hepatitis and cirrhosis, and it's thought to be the cause of about half of the hepatocellular carcinoma cases worldwide. However, unlike HCV, Hep B is cleared by the immune system in the vast majority of cases. Only about 5% of infected adults end up with chronic infections. But this virus is so widespread, so many people get infected, that it's estimated there's about 400 million chronic HBV infections worldwide. So that's pretty staggering. And the hepatitis B genome encodes 7 proteins. Only one of these, HBX, has been linked to cancer. So that one protein, HBX, has several different effects, can increase cell division, can interfere with some known DNA repair mechanisms, and it also blocks the activity of this P53 protein, whose normal job is to activate apoptosis in sort of precancerous, damaged cells. And what's kind of interesting is in most HBV infections, the genome is maintained as an episome, meaning it's not integrated into the host cell chromosome. But in a very high percentage of the HBV-associated hepatocellular carcinomas, we do see the viral genome integrated. And so that increases the typically it's believed to increase the transcription of the viral genes. So for example, you would likely be producing more of this HBX protein in infections where the viral genome is integrated, and so you would be getting more of these undesirable activities. Luckily, there is an HBV vaccine, so that was actually technically the first anti-cancer vaccine developed, right? If we can prevent people getting the virus, getting these chronic infections, we can prevent these cancers as well. The second small DNA tumor virus is this attributed to this Merkel cell polyomavirus, which is actually a very common virus. About 80% of adults are believed to be infected by the time they are 50 years old. But it is linked to a rare and unfortunately aggressive skin cancer. So Merkel cells are these epithelial cells that are linked with touch perception. And really, we tend to only see this cancer developing in people that are either very old and or immunocompromised. So if the immune system is not functioning at capacity, it seems the increased viral load is able to kind of tip the scales in favor of cancer. And polyomaviruses do produce these what are called T antigens, stands for tumor antigens. There's a large T and a small T antigen, but together these actually help promote pushing the cell past checkpoints into S phase, which is where DNA replication happens, and also where you would get some of the enzymes available that you need to replicate the viral genome as well, right? So there would be some advantage to the virus to getting the cell into S phase, but it just that push past that checkpoint is believed to be enough to help push those cells towards cancer, especially in immunocompromised individuals. And then the third example here of small DNA tumor viruses linked to cancer is HPV, which if you've heard of any of these, you've probably heard of this one. So it's the most commonly common sexually transmitted infection in the U.S. These are small icosahedral non-enveloped viruses, got circular genomes, and they tend to infect either basal layer of skin cells or mucosal epithelium, and they're best associated with warts, so typically genital warts. There's a lot of different strains of HPV, over 200 that have been identified. So there's some low risk types, meaning low risk for cancer, but they can still cause other issues. And then there's some high risk strains. And the highest risk strains are 16 and 18. So you detect those one or both of those in almost all cervical cancers. But that being said, that there are some other strains that have been linked to cervical cancer as well. And you see a picture of a pap smear here, right? So early detection test for cervical cancer, where you're literally just scraping off some cervical cells, smearing them on a slide, and taking a look to see if you see any abnormal cell morphology that could be an indicator of precancer. Very effective screen. And so normally you hear about HPV in relation to cervical cancer, and certainly that's important, but there's also a number of other different cancer types you can see here where HPV is a major contributing factor. So a lot of people may think of this as, okay, well, this is causing cancer in women, but it's also causing cancer in men as well. So a pretty high percentage of these oropharyngeal cancers are actually men. So in terms of how HPV causes cancer, the HPV genome actually codes two oncogenes, this E6 and E7. E6 will block this P53 tumor suppressor gene, which would, as I mentioned a minute ago, would be, its normal role would be to trigger apoptosis and damage cells, so it's blocking that from happening. E7 is interfering directly with retinoblastoma, which, as we talked about earlier, right, is that red light one of these cell cycle checkpoints. So if we interfere with that, we're basically turning the red light green. So we got these two activities. And again, this seems to be a case where in those highest risk HPV strains, the viral genome is actually integrated into host cell chromosome, and we get excess production of these oncogenes, and that seems to be linked to cancer. Luckily, there is a HPV vaccine. There have been a couple of formulations over the years. First introduced in 2006. It was called Gardasil. Originally it was just recommended for sort of preteen females, but now it's recommended for both males and females, really up into the early 20s, early to mid 20s. So these are recombinant vaccines. Basically the vaccine is the capsid protein of the HPV virus. And there was a newer formulation came out just a few years ago, 2014, and it included some additional strains, so it gives you a bit broader protection. So we have seen pretty good adoption of Gardasil, especially in more recent years, but this could dramatically reduce some of these specific cancers. All right, then our last group, the large DNA tumor viruses, includes two, Epstein-Barr virus and Kaposi sarcoma virus. So these are both herpes viruses. These are large, enveloped. They've got what are said to be complex double-stranded DNA genomes. And herpes viruses are, they always say herpes is forever, right? So these are viruses that once they've established their infection, they can go into what's called latency, where they basically will go through these extended sort of hibernation periods where they're in the cell, but they're hiding out from the immune system, and then they can come roaring back temporarily, and then go back and hide out again for a while. So let's talk about Epstein-Barr, EBV first. So it can affect both B cells, immune cells, and epithelial cells. And it's best known for its link between certain types of lymphoma, so Burkitt's and both Hodgkin's and non-Hodgkin's lymphoma. And that's due to its ability to infect these B lymphocytes. But it can also be linked to a couple of different types of carcinomas through its ability to infect epithelial cells. So yeah, here you see this poor kid with lymphoma, and his lymph nodes are so swollen and distended. It looks incredibly uncomfortable. So interestingly, especially in the terms of the lymphoma, there was this link between immunocompromised individuals and the development of cancer. So actually Epstein-Barr virus, a high percentage of the population is infected with that virus, but we tend to see these cancers developing primarily in immunodeficient individuals. This link was first made in Africa, where it was observed that we were seeing this lymphoma rise in regions where malaria was really prevalent. So the malaria infection was knocking down people's immune systems, and then the Epstein-Barr virus infection was triggering these lymphomas. And I'm not going to get into all the details here, but these viruses have pretty large genomes, and they go through what are called these latency stages, which are basically these kind of different transcription programs where different genes are being activated at different times during the infection. And really the main thing I wanted you to take away from this table is that in a number of these different latency stages, right, there's various genes that get transcribed that all have cancer-promoting activities of one variety or another. So again, lots of mechanisms by which this virus could help drive cancer. And we see something similar in Kaposi sarcoma virus, another herpes virus. In most individuals, it causes subclinical infection, meaning no symptoms. But this really first came on the radar during the AIDS epidemic. So it was seen that HIV positive individuals that were progressing to AIDS, immunodeficiency, were developing this rare skin cancer, Kaposi sarcoma, associated with this particular virus. You can see a picture of what that looks like here. So it seems to be another instance of in immunocompromised individuals, the virus is able to proliferate and transcribe at levels much higher than it would be in somebody with a fully functioning immune system. And that seems to be, again, tipping the scales in the favor of cancer. So although you usually think of Kaposi sarcoma associated with this virus, it can also be linked to a couple of different rare types of lymphoma as well. Oh yeah, I was going to mention this too. So, another place you typically see people having their immune systems compromised is following organ transplant, right? So you're intentionally suppressing the immune system to try to prevent rejection of the organ. So we also see a much higher risk of Kaposi sarcoma in people that have had an organ transplant. And very similar to what we talked about with Epstein-Barr virus, with Kaposi sarcoma virus as well, we have these latency stages, or lytic versus latency stages, and again, just a whole host of genes, I won't go through them all, being transcribed that have cancer-promoting properties. Okay, so not entirely surprising given this list that you would potentially see cancer being associated with this particular virus. So anyway, I know there's a lot of information in there, but hopefully that gives you some feel for what are the cancer-causing viruses in humans, and what are at least some of the mechanisms by which they can cause cancer.